I would like to point out that 2 independent labs had also used DFMO in a slightly different model. It's a transgenic model, that a mouse that has neuroblastoma or gets neuroblastoma by overexpression of NMEC. And they use DFMO in different combinations, not etoposide, but again, I cannot discuss all the data, but they published their research in cancer research. Mike Hogarty from the Children's Hospital in Philadelphia, who approached me in around 2005 when our oncogene paper came out and asked me about polyamines, and he started to get interested in it, so it's wonderful. John Cleveland, previously at St. Jude's, now at Scripps, Florida, he also, almost just 6 weeks later, published another study with the same mouse model, also with DFMO combinations. So clearly there started to be interest in this area around 2008, 2009. So then Dr. Scholler decided that we should write up this protocol, clinical trial protocol, and basically there's now 3 hospitals, from first in Vermont, now here in Van Andel, and in Florida, in Orlando, and also the Children's Hospital of Orange County in California is involved. And we hope that at some point also the Kapiolani Medical Center in Honolulu can be involved in the trial. So what was the primary objective of this trial? It's to determine the safety, as we just heard in the previous descriptions of clinical trials, and tolerability, as well as MTT of DFMO as a single agent, as well as in combination with etoposide in the pediatric young adult patients with refractory or recurrent neuroblastoma. The secondary objective was to evaluate, or is to evaluate, the activity of DFMO as a single agent and in combination with etoposide in these tumor types, based on progression-free survival as well as overall response rate. To evaluate the pharmacokinetic parameters of DFMO as a single agent, and then as well also do all the biological studies, which is really where I come in, and my lab comes in, and also Dr. Gerner's lab at the University of Arizona comes in to evaluate the biological samples for ODC activity and polyamine levels before and after treatment of the patients. This is the layout, looks very similar to the previous clinical trial that we just heard. We have the initial evaluation with DFMO and twice daily for a 21-day cycle. Then there's the re-evaluation, and there's also a dose escalation study, 3+3, where 3 patients are being treated first at 1 dose level, and then if there are no adverse effects, we move on to the next level of dose, here 750 mg per square meter. In the second cycle, through the 5th cycle, then etoposide at 50 milligrams per square meter is being added for 14 days and then removed for 7 days, but DFMO remains in the regimen. So this is the general layout of the trial. So in summary, in the preclinical data, we found that DFMO rapidly inhibits neuroblastoma cell proliferation in neuroblastoma cells by inducing the G1 cell cycle arrest and activating the P27RB pathway and also MYC and downregulation. In the in vivo mouse models, in the xenografts model, as well as in the transgenic mice from different labs, DFMO was effective in prolonging the tumor-free survival. And finally, this old drug that has been around since 1978 has been repurposed for this particular trial. And based on the current lack of effective therapies for these patients and for its preclinical effectiveness, as well as its high safety profile, we then, as I now just explained, to advance DFMO to the clinic. Just 2 more slides on what we're doing right now, future. We actually look at various combinations with DFMO, including 940-MOX, Solindac, topotecan, bortezepamine, and doxorubicin, for example. These are just some samples. We're looking also at HDAC inhibitors, as Carol Thiel just spoke about, AKT inhibitors, mTOR inhibitors, as well as ALK inhibitors. And we also have an interest in the polyamine transport inhibitors because sometimes cells have the ability to take up polyamines after you deplete them intracellularly. So that means they can replenish their polyamines, and that can be a reason why some of the cells don't always respond as effectively. And so the goal will be sort of to plug up those pumps that bring in those polyamines from outside, from the extracellular environment, And there are certain inhibitors that have been developed here, like AMXT15-1 from Aminex Therapeutics. And Mark Burns, who is here in the audience, has provided us with this compound, and we're planning some xenograft studies to do that together with Dr. Scholler. And we're looking at some other polyamine inhibitors as well that inhibit other parts of the pathway. Another approach, and that's my last slide, is a completely novel and somewhat proprietary type of nanoparticle that we have devised together with an assistant professor in pharmaceutics now in my new department where I am. And basically what it is it's a nanoparticle that is different from previous nanoparticles that is expressing antibodies and recognizes the epitopes such as GD2 in neuroblastoma. And let me see if my little clip works. These nanoparticles then attach to the GD2 epitope, and this will be DFMO, and we can even have a second drug in this particle. And what happens is the particle fuses with the membrane, and the drug goes inside, and hopefully the drug has its effect on the cells, and the cells die. So it's just a more targeted delivery of DFMO, basically, rather than just giving it by itself. With that, I would like to close. This is my group as it was maybe in 2010. Please note that February 2010, everybody's wearing shirts, short sleeves. Dr. Kawamoa has been with me for 5 years, and she just received the tenure-track assistant professor position, and so she will no longer be working for me, which is horrible, because she has been great, and I don't know what I'm going to do without her. Chris Wallach was a bachelor student, but unbelievable. He went on to do PharmD program, and he's a very bright guy. He was the first author on the oncogene paper. And there's a series of other students that have contributed over the years. Collaborators, first and foremost, Dr. Scholler has been great to work with, and I look forward to continuing to working with her in the future. Dr. Maskens, of course, from UC Irvine, has been over the years, he's a polyamine researcher, and he's been very important in my career because he has helped me as an almost external advisor, if you will, to move through my career. And he's obviously also involved in the clinical trial. He has done sentinel work with DFMO in the chemoprevention arena, preventing colorectal cancer after polyp was removed. And Dr. Jean Gerner, who is a strong collaborator with Dr. Maskens at the University of Arizona, who are also both co-founders of this company, Cancer Prevention Pharmaceuticals, that is making or providing us the DFMO. Tony Peck from Penn State University and David Feith were both polyamine collaborators. Dick Gertz from the University of Amsterdam produced some of the data for us. Patrick Wooster is the person that gives us the DFMO that is not being used for human studies, but for animal studies, and he has a lot of it for that. Bob Casera from Johns Hopkins and Craig Bias and Michael Perron were also people that contributed. I received, and I'm very thankful for that, funding by an R1 grant that I have from the National Cancer Institute. And without that, this work also probably could have not taken place because it was based on studying polyamines in MECAN-amplified neuroblastoma. And I had several supplements as well as an ARA grant. And then the Hawaii Community Foundation is a smaller organization, but provides some nice $50,000 to $100,000 grants. That they were instrumental in pushing that project forward for me on the preclinical level. And Magic Water Foundation also provided us with funds to do some combination studies, which was much appreciated. This is the lab today. Faces have changed, some of them. Dr. Cuomo is still there. And this is it. Thank you very much. This is an article from a newspaper reporting on an old drug that has new promise to fight cancer and talking about the neuroblastoma project. And I was lucky enough to win the inaugural 2010 Bachmann Award for Innovation and Translational Research, and that produced that little newspaper article, which was very nice. So thank you very much. Thank you.